Hello, and thank you for joining us. In this video, we'll explore results from DeepBio's AI model developed to assist with the detection of breast cancer metastases in formalin-fixed, paraffin-embedded sentinel lymph node sections. This model is designed to support pathologists by highlighting regions with a high likelihood of tumor presence, including isolated tumor cells, micrometastases, and macrometastases. These distinctions can play a key role in assessing disease spread and guiding treatment planning. The model aims to aid in the identification of subtle and scattered tumor foci that might otherwise be overlooked. Let's take a closer look at how the model performs across a range of real-world slides. We begin with a high-level overview showing 5 formalin-fixed, paraffin-embedded sentinel lymph node slides. These thumbnails provide a snapshot of the AI's results, which we'll walk through in more detail. The first 2 slides represent micrometastatic involvement. The related patches will be displayed at 100x magnification to visualize the smaller tumor foci better. The patches of the remaining 3 slides that contain macrometastases are presented at 50x magnification. Regions highlighted with a red overlay indicate areas with a high likelihood of tumor detected by the AI model, which range from isolated tumor cells to dense tumor clusters. Let's start with case 9266539, where micrometastatic disease was observed. The zoomed-in images on the left and right show each region in greater detail. In the first region, the model detected 3 small foci suggestive of tumor cells. In the second, a larger tumor cluster is highlighted. Here, we take a closer look at the first region. The image on the left shows our AI results, while the image on the right is the original whole slide image for direct visual comparison. The model highlights 3 small, well-circumscribed tumor foci within adipose tissue, all features suggestive of micrometastatic involvement. This level of detection, particularly in adipose-rich areas where small foci can be overlooked, may assist with more consistent interpretation during the nodal assessment. In the second region in the same case, the AI highlights a larger tumor cluster with clear boundaries, providing a visual cue for review. This example features 2 very small tumor foci, identified by the model in adipose tissue, measuring approximately 104 and 65 micrometers. The model's ability to flag such small foci highlights its potential utility in reviewing slides with low-volume metastatic involvement. Here, the AI highlights a likely micrometastasis alongside a much smaller focus resembling isolated tumor cells. Both regions are clearly outlined, showing sensitivity to a wide range of tumor sizes. In this example, the AI flags 2 distinct tumor foci along the lymph node margin, an example of detection in the peripheral regions. In this example, the AI correctly identifies the main lesion but undersegments part of the focus. Despite the slight miss, the central tumor region is clearly captured and localized. This next case includes 2 regions of micrometastatic involvement, flagged here and presented in the context of their full slides. The AI detected a long, narrow micrometastasis near the capsule. The AI accurately follows its contour despite its irregular shape and alignment with the surrounding stroma. Here, despite a scan artifact and image blur on the right, the AI still highlights the key tumor region, illustrating robust performance in less than optimal imaging conditions. 2 small foci, measuring around 63 and 72 micrometers, were detected here. This reinforces the model's sensitivity to small volume disease. Despite staining artifact along the margin, the model correctly identified a focus suspicious for cancer, showing robustness under variable slide quality. In these examples, the model captures one of 2 tumor clusters, missing a nearby smaller focus. However, the primary lesion is well localized. These cases highlight ongoing opportunities for fine-tuning while demonstrating consistent detection of dominant features. Moving on to the next case, the model has flagged 2 macrometastatic foci. The left and right panels provide zoomed-in views confirming extensive involvement. Here, the model accurately captures the full extent of this macrometastasis, which occupies a large contiguous area of the lymph node. In addition to the main tumor mass, the model also identifies smaller adjacent clusters, showing good performance across both high volume and scattered patterns of involvement. This is another macrometastatic case. The AI captures a dense, centrally located tumor cluster with good contouring, despite variable staining in the surrounding tissue. The model accurately detects and outlines the macro focus, even with pale staining and a densely cellular background. That concludes our review of the sample slides. These examples illustrate the model's ability to detect both macro and micrometastases, even in challenging conditions like variable staining, tissue artifacts, and small or irregular tumor clusters. We look forward to your feedback and any discussion on how we might support further evaluation or integration into your workflow. Thank you for watching.